So it is in fact 3:10, and I'm sure some people will trickle in, but I will go ahead and start the day, I guess, or start these sections. I'm already a little lost in my Zooming, but that's okay. We are now in our sixth talk of our 20 talk series for senior experience in the chemistry and biochemistry um, talk series, I guess. See, my talking is very bad. So I'm really happy that you can join us here for Galini's talk, and I hope that you will have an opportunity to join us for Katie's talk, which is directly after this talk at 4 o'clock. We will continue our series next week with 5 talks next week, starting on Tuesday. We're going to have one over the convocation hour, or 12 to 12:50 on Tuesday, starting with Winnie. So we will see you then, and you can expect a series of emails from me to remind you maybe about these talks if you are from outside 680. If you are in the 680 class, you already know. I'm going to pass it off to Kim to do these introductions. So here's my introduction to Kim Dixon.
Thank you, Deanna. Galini, I was just awarded your advisor position just this year. But luckily I got to know you last year in molecular biology a little bit. That's a big class. So I was really happy that I got to know you more in advanced biotechnology last year, where it was really fun to see your personality and how dedicated you are to your science. I remember how eagerly you dove into the primary literature and left no stone unturned when it was your turn to lead discussion and choose papers. So that enthusiasm and dedication and determination was really appreciated. But it was also fun to get to know you as a person and see your sense of humor. Hear your witty jabs at Nabor occasionally in the lab, all those good things. And just see your just embracing research in particular too. So I was thrilled that you were able to go spend the summer in Dr. Kramer's lab at Dartmouth, and I'm sure you were just a productive and very welcome asset to their lab over the summer. And you came back so eager for graduate school and more research science in your future. So I look forward to hearing your talk today, and I look forward to seeing where your science trajectory brings you.
Thank you so much for your introduction. So yeah, today I'm gonna begin my senior experience talk with investigation of beta blockers as a potential antifungal drug against Aspergillus fumigatus and Fusarium solani ocular infections. As Kim said, I had the pleasure of working with Dr. Robert Kramer, who's an Loras alumni in Dartmouth College, and Kimberly Dixon and Deanna Donahue have been my advisors over this 1.5 past years. So I know this title can be very overwhelming, which is why I want to break down what I'm looking for. So when I talk about ocular infections, the term ocular is used, when it pertains to vision or any component of the eye, and infections are caused by microorganisms that invade the body. So when I talk about ocular infections, I'm specifically talking about microorganisms that invade the eye. So a little motivation for you for why you should care and listen to this talk. Are eyes really that important? Well, yeah, we use them every day, and I think you already knew the answer to that. But are eye infections that common? Surprisingly, they actually are. According to the Academy of Ophthalmology here in the US, there's nearly 1 million eye infections, most of which actually come from contact lens use. So an example of an ocular infection outbreak in the US that came from contact lens solution use is this one that had to do with Renew with Moisture Lock, which is a specific brand of contact lens solution. And we can see here it started all the way in June, and the number of cases of ocular infections increased. And then when the specific product was withdrawn from the market, the number of cases fell back down. And these patients were from all over the US, from 33 different states. But what was the most concerning to us was that over a third of these patients needed to have a corneal transplantation, which I'm later going to explain what that is. And the specific ocular infection outbreak was an outbreak of fusarium keratitis, which I'm also going to explain what that is later on. So the ocular infection of fusarium keratitis is an infection of the cornea, and it's caused by fungi, filamentous fungi of fusarium, and they almost always threaten sight, and they can even result into vision loss, which is why we need a corneal transplantation. So what a corneal transplantation is that this is what your eye looks when the specific fungi has invaded it and caused an ocular infection, and then you remove the first layer of the cornea, and then you basically stitch on new donated corneal tissue. And this is an example of what fungal keratitis looks like. This dry necrotic surface is very indicative that there's a fungi that caused this. So this is what it looks like in real. So what specifically causes fusarium keratitis are 3 species. First is the Candida species, the Aspergillus species, and the fusarium species. But we're specifically going to talk about fusarium species because they make for over 50% of fusarium keratitis cases. They are specifically solid fungi, and they're pathogen to plants and humans equally. Some examples of plants that they can invade is rice, weeds, oats, and of course they also invade humans, which is my focus specifically. And the species that I'm going to focus on is Fusarium solani. This is how they look like under the microscope. They have a very characteristic banana-shaped conidia, so you can see how they look like. And here, this graph basically is a summary table that shows how prevalent ocular infections caused by Fusarium species are. So here, all the way on the left, we have different locations where ocular infections happen. So we have basically from every single continent. And then this is a number of patients or subjects who were part of the study. And then this is a number of patients who tested culture positive. And here we see what specific species caused that ocular infection. And as you can see, we have Aspergillus, Candida, and Fusarium, but the majority of them make Fusarium. So, it basically reiterates that Fusarium is the most common causative agent of Fusarium keratitis. So some factors that affect the prevalence of Fusarium keratitis have to do with climate. So it has been shown that in tropical climate, Fusarium keratitis outbreaks happen more often, and they also have to do with the temperature. So atmospheric temperature and humidity. The higher they are, there's a correlation with a higher number of Fusarium keratitis cases. So that's very interesting as well. And if you work in the agricultural sector, and specifically if you have a lot of access to the fields and you work during harvest months, you're more prone to the fungi invading your eye because, again, this fungi is a soil fungi, so it can attack you more easily. Of course, some other factors that affect the prevalence of Fusarium keratitis have to do with the history of the patient. So if you have past ocular trauma, or if you're an immunocompromised individual because, for example, you have HIV, that again makes you more open to getting a Fusarium keratitis. So the current antifungals, which are drugs to treat fungal infections against Fusarium solani, are azoles and polines. These are 2 examples. One example for azole and one example for poline. So azoles are very characteristic compounds. They're a class of 5-membered heterocyclic compounds that contain 1 nitrogen atom and at least 1 non-carbon atom. So again, we see 2 nitrogens here. And then polines as drugs are a polyunsaturated organic compound that contain at least 3 alternating double and single carbon bonds. So this is how polines look like typically. Some other examples of drugs are clotrimazole, iconazole, and itraconazole that fall under the azole category, and then amphotericin B that falls under the choline category. So how exactly do these antifungals target fungi and help them treat them? Well, the targets are also to do with the cell wall. So one way that you can target the cell wall is through the cell membrane. And because the plasma membrane, serves as a barrier to the inside of the cell, the fungal cell. The second way is through targeting sterile biosynthesis. So sterile is a very vital component of the cell membrane. So again, when you target that, the production of sterile, you basically weaken the cell membrane and you can attack the fungal cell more easily. And lastly, you can also target beta-glucan biosynthesis, which again, it's one of the most abundant polysaccharides that are found in the fungal cell wall. So again, they all have to do with either the cell wall or the plasma membrane of the fungi, because those have been proven to be hard to get past to attack the fungal cells. So specifically, I want to talk about how azoles and pollens work. So here, as I talked before, azoles target ergosterol. So usually ergosterol happens from lanosterol getting converted into ergosterol, and what azole does is that it targets that formation, and instead of having ergosterol, you have a big accumulation of lanosterol, which is very toxic to the fungal cell. So that's how azoles work. And then amphotericin B or pollens, they interact with the cell membrane here, and they form channels through which molecules from the inside of the cell leak to the outside. So you kind of break that barrier that protects the inside of the fungal cell, and you can attack it more easily. And lastly, a third type of drug that I didn't mention before is echinocandin, which target beta-glucan synthase. So when you stop the beta-glucan synthase production, that weakens the cell wall, which again makes these drugs effective. So the problem with these curing drugs that we use is that they have shown, that Fusarium has shown a very high resistance to this drug. So as I said before, drug resistance is very common, and approximately 1/6 of a subject that have Fusarium keratitis experience a corneal perforation, which means that the cornea has been penetrated and it leaves the cornea very damaged. And that leads to corneal transplantation. So this progression of Fusarium keratitis actually happens over a week, and that's because the curing drugs that are used for treatment aren't as effective, and because if you cannot correctly identify the species that caused Fusarium keratitis and you give the wrong drug, that will not be effective at all. So this is an example of how virulent Fusarium species can be and cause Fusarium keratitis. So I want to talk about the taxonomy and taxonomic hierarchy of the Fusarium species. So they're obviously Eukarya and fungi, and then you go all the way down to phylum, class, order, family, the genus Fusarium, and then the species Fusarium solani. But even within the species, you have so many strains, and each strain has a different antifungal susceptibility. So one strain might be treated and cured with the drugs that we already have, but other ones may not. And that's something that you're gonna also later realize, but I just wanna focus on that as well. So the current fungal keratitis treatment is 5% topical natamycin, which is the only FDA approved drug. And they work like eye drops, so you just put them in your eye. And it's actually been proven in the mycotic ulcer trial treatment, which is a very famous, trial treatment in the ocular infection category, that natamycin was more effective than voriconazole, specifically in Fusarium infections. But what came out of that study that was really interesting and kind of propelled us to this specific research is that we saw synergistic interactions for natamycin and voriconazole with non-antifungal agents like 5-fluorouracil and beta-blocker timolol. So this was the idea for our project, because we already know that there's a lot of drug resistance within the different strains of the Fusarium solani species. and that can increase, these vision-threatening infections. And we already know that just using one drug doesn't always work. So that made us want to look not only into non-antifungals that can be used as antifungals, but also into combination therapy. So the potential drug that we wanted to look into that's not an antifungal but can be used as an antifungal are beta blockers. And we specifically thought that beta blockers could target the calcium pumps that you can see here are in the cell membrane. So usually calcium pumps, they're there to control homeostasis between the intracellular and extracellular components of the fungal cell, and free calcium regulates multiplication, transportation, elongation, and growth. So when you target these calcium pumps, you basically ruin that balance that the cell has, and that's our thinking of how beta blockers could work in fungi. So beta blockers are usually used in human health for cardiovascular and non-cardiovascular diseases. And different beta blockers have different degrees of lipophilicity. So that makes them cross the blood-brain barrier in different ways. And I mentioned that, and you may be wondering why lipophilicity matter, like why does it matter? And why do we wanna find more about it? So lipophilicity is the ability of a compound to dissolve in fats, lipids, and nonpolar solvents. In humans, that has to do with our blood-brain barrier. In fungi, that has to do with the cell wall and the cell membrane. And the lipophilicity of a drug, whether it's an antifungal or a non-antifungal, can impact the solubility, permeability, and potency, as well as toxicity. So it has been shown, for example, in humans, when we use beta blockers that have high lipophilicity are more permeable and more potent, so they can cross our blood-brain barrier more easily, but because of that, they're also more toxic. So we're gonna take that idea of the high lipophilic beta blockers that can permeate the blood-brain barrier to fungi and see if that translates from humans to fungi. So some of the questions we wanna answer is if the antifungal property is shared across all beta blockers, because as I said before, there's lipophilic ones, but also, hydrophilic ones. If there is a more potent beta blocker that we need to focus on, and whether lipophilicity has to do with antifungal property and, how strong the beta blocker can be. And if there is a human beta blocker receptor in fungi, because we already know that in humans, the beta blocker receptors are beta 1 or beta 2. Beta 1 is usually in the heart and beta 2 is in the bronchi, but we don't know something, accordingly in fungi. So we want to find more about that so we can target them more easily. So these are the beta blockers that I'm going to mention throughout my talk, or I'm going to use in my research. So our current hypothesis is that lipophilic beta blockers are more effective because they're also more effective in humans in crossing the blood-brain barrier. And so for high lipophilicity beta blockers, I'm using propranolol as well as timolol. And then for medium lipophilicity, we have nadolol. And then lastly, for low lipophilicity, we have atenolol and sotalol. So these are beta blockers that I'm going to mention at some point throughout this presentation. So first, before we see how effective these beta blockers are, we have to measure how effective they are. And to do that, we have to find the MIC, or minimum inhibitory concentration. And we do that by using a 96-well plate. And so vertically, so in different columns, we have the concentration of the drug we're using in increasing concentration. So again, we start from a low concentration of drug to a high concentration of drug. And then we use one column just with fungus. As a control. So here we expect to see a lot of growth because the fungus can grow by itself. And then here in column 12, we only have media. So we expect to see no growth. And then going horizontally, we have the fungal cells. So we usually want to test one strain per 2 rows. So here there would be strain 1, strain 2, strain 3, and strain 4. And the strain concentration is the same throughout the entire cell besides column 12, because that's only media. And so how we report MIC is by comparing the media column where there's no growth to the column that shows the least growth. So an example of that, I found a picture. It's not the best, but as you can see here, this is the media column and there's absolutely no growth. It's very clear. And the MIC is in column 1. So it would be the concentration of column 1. Let's say, for example, here it was 250 micrograms per milliliter. And you can see in between the varying growth levels, and that has to do with, the concentration of the drug and how it affects the fungi. It's also very noticeable. Sorry, this is where the MIC is. It's also very noticeable how different strains grow differently. And that's something that I mentioned before. We cannot predict how each strain will not only grow, but react to the drug. For example, this specific strain in CND has a pretty high virulence, as you can see. It's pretty cloudy here. And that's the issue with treating these fungal infections from Fusarium keratitis. They vary so much between strains. So first, I tested beta blocker propanolol as an antifungal. And if you remember, that's the most lipophilic one out of all of them. And so I had different strains of Fusarium keratitis. And I tested propanolol R, because propanolol is a chiral compound. So I tested R as propanolol and then the combination. And the MICs that I found were actually very comparable to the MICs of natamycin, which is the first line of treatment for Fusarium keratitis, if you remember from before. So that's a very promising result, that beta blockers can be used as antifungals and be as effective as natamycin, which is an actual antifungal. Then I tested nadolol, atenolol, and sotalol, and unfortunately, I did not find an acceptable MIC for these 3 beta blockers. If you remember, nadolol was the one with a medium lipophilicity, and atenolol and sotalol were very hydrophilic, so very low lipophilicity. And when we say no MIC, we mean there was so much growth that not even a high concentration of the drug could kill the fungal cells. And when we test the different concentrations of drugs, we always have to make sure that we don't put like an outrageous drug concentration, because if it works in fungi, we need to be able to translate it in humans. So I can't just put a very highly concentrated drug into a person. So that's always something we have to remember. And so propanolol and nadolol, they're both very similar in structure, as you can see. Nadolol has this additional OH groups here. And they're both first generation beta blockers. So that means that they both target beta 1 and beta 2 receptors, but only propanolol had antifungal properties, which makes us believe that lipophilicity does play a very important role in targeting and killing these fungal cells in Fusarium keratitis. As I said before, we also want to look into combination therapy, so not only using one drug, but a combination of drugs. And this is a summary of a study that was done for Fusarium keratitis that investigated combinations of drugs. So here we see they use natamycin and other pollins in combination with azoles, and they did see synergistic effects for that specific drug combination. And again, all these Fusarium keratitis cases were a result of Fusarium species invading the eye. So this was kind of the background for us wanting to look into combination therapy, but not only between antifungals, but also between antifungals and non-antifungals like beta blockers. So how we find whether there's a synergistic interaction between 2 drugs is by finding the FICI, which basically utilizes the idea of MIC. So FICI is a formula that you basically have to find the MIC of drug A in combination with drug B, and then put that over drug A's MIC by itself, and then drug B with drug A over the combination of over the drug B's MIC alone. So that's how you find whether there are synergistic interactions. And so here are some results for FICI for Fusarium species, where we had a combination therapy, so 2 different drugs. So here we have Fusarium solani causing the infection, and we tested natamycin with a non-antifungal, which is 5-fluorouracil. This is not a beta blocker, but I just wanted to show that besides beta blockers, there's also been testing in other kinds of drugs. I think 5-fluorouracil is used in cancer, and in some strains we did see some synergistic interaction. But then TIM is timolol, which is a beta blocker, and again it showed some synergistic interactions for Fusarium solani species. And again, we tested that for voriconazole and timolol and 5-fluorouracil, but we didn't see any synergistic interactions. You can find if there's a synergistic interaction between two drugs if the FICI value is less than 0.5. So as you can see, all the bolded values are below 0.5. And something else that I also want to emphasize in this table is that different strains react differently to these drug combinations, which again, I feel like I'm repeating myself, but again, it just shows that different strains react differently, and that's why it's so hard to treat these infections. So this was kind of the background of using beta blockers like timolol and natamycin to find better combination therapies. And so what we did is use other beta blockers in combination with other drugs. So we tested nadolol and natamycin, but we found no synergistic interactions between the two drugs. And if you remember, nadolol is the one that has a medium lipophilic level. We also tested the Fusarium isolates with sotalol and natamycin, and we didn't find any synergistic interaction between the two drugs either. And we also tested atenolol and natamycin, and again, we didn't see an acceptable FICI. So again, there was a lot of growth, and using these two drugs together didn't work, or didn't work better than just using natamycin alone, because natamycin works anyways by itself. So again, here, I basically wanted to show a lot of data in a concise way, but hopefully this is not too overwhelming. Each table kind of represents the 96-well plate, where RPMI is my media. Here we only have fungus, and then everything that's green, it means that there is growth, and everything that's black, there is no growth. And so here I tested nadolol and natamycin together for different strains. As you can see, these are different strain numbers. And then I used atenolol and natamycin again for different strain numbers. And if you couldn't already tell by the FICI values, unfortunately, we didn't find any synergy between natamycin and nadolol because all these values are above 0.5. And you might be thinking, why? Because there's no growth where there's black. So that means that, the drug must have worked. But unfortunately, the effect, the result of killing like all the fungal cells isn't because we used a combination of natamycin and nadolol. It's only because of natamycin. Natamycin works by itself anyways. We already know that it's the first line of treatment. So we didn't learn anything that we didn't already know, but it kind of basically reiterates that lipophilicity does play a role, because only propanol has been shown to work so far. Again, this is for different strands with nadolol and natamycin and atenolol natamycin, and the FICI values that are over 0.5 show that there is no synergistic interaction between the two drugs. And something that you may have observed here is that there's a different number of like black cells here, and that's because, again, each strain reacts differently to the drugs that we have. But again, all the black cells are because natamycin was working, not because natamycin was working with nadolol or atenolol. These are example of well photos. So I took photos of the little wells, and this is what it looks under the microscope. So here we have 8 micrograms per milliliter of natamycin, and then 16. You can see the filamentous nature of the fungi here, and this is like, there is some growth, but there isn't too much. But this is the MIC, so 16 micrograms per milliliter of natamycin for this specific strain is what kills the entire cell population, and you see there's no growth at all. And And when we compare that for the same strain of 060110, we have here positive control. So you see there's so much growth. And here we have atenolol, which again, there's less growth than, just the fungi, but there's still like a very big amount of growth. And here we see when we combine natamycin and atenolol, there is like very, very few growth, but that's because natamycin is doing most of the work and not atenolol. So what we've learned so far is that propranolol works, and specific for Fusarium keratitis species, and specifically the MIC that we found for propranolol is 31.25 to 62.5 micrograms per milliliter. And again, this is the range because different strains react differently. Timolol, in that background paper that I showed, did show synergy with natamycin. Nadolol wasn't here, so just imagine it's somewhere there in the middle. It has medium lipophilicity. It didn't work by itself or in combination with other drugs, and neither did atenolol or sotalol. So again, there was no MIC detected for the drug, for drug concentrations that were less than 200 micrograms, which is a pretty high amount. And again, there was no FICI with natamycin, which is our current first line of treatment for Fusarium keratitis infections. So the degree of lipophilicity does matter because, as we see, highly lipophilic beta blockers had the ability to have some kind of result against Fusarium solani species, but unfortunately nadolol and atenolol and sotalol did not work. I would love to test more, beta blockers like carvedilol or metoprolol, who are also, on the higher level of the lipophilicity spectrum, to see, if they show some kind of acceptable MIC that we can use also in humans. But yeah, the lesson of the day is that the degree of lipophilicity does matter for beta blockers, and we have some promising results for propranolol, but not so promising results for atenolol, sotalol, and nadolol. So that was my research that I did over the summer. I want to thank, obviously, Dr. Robert Kramer for accepting me in his lab, but I also want to thank Dr. Diana Donahue for helping me so much to make this presentation, and Dr. Kim Dixon for being such a good advisor and taking over and helping me. And of course, all my teammates, Evan for borrowing me his microphone, and everyone who's here watching me. So yeah, I want to thank you for being here. That's it.
Yay!
Thank you so much. I will open the floor for questions, so you can unmute yourself and ask a question. You can raise your hand and have me call on you, or you can even chat a question that says, I would like to ask a question to me or to General. Question?
Yes, Kate.
So what are the next steps? Like, what are the future plans of this research in this group? Where are they going after this?
Well, I obviously want to test more beta blockers and, especially more highly lipophilic beta blockers, but I also want to test out more strains. And we're also thinking of using a specific gene that we know regulates stress in the cell membrane of Fusarium keratitis, and see how, if we knock down that, how the cell reacts. So there's definitely a lot of work to do, but there's not enough time. But yeah, that's pretty much it.
Other questions?
Okay.
So I have two questions. One is, you tested drugs against a bunch of different strains, and you talked about how those strains are different. I was just wondering if any of the fungal strains that you worked with count as things that are particularly resistant to, the drug resistant, like you would see in a, sort of in a bad pathology situation, or if those are sort of more wild type strains. And my other question was that, does the FICI value of 0.5 usually translate well into like a medical situation. So if you observe that value of less than 0.5 in vitro, does that also generally translate well into a therapeutic treatment?
So for the first question, I know that all my strains came from ocular isolates of axial infections, because the PI that I worked with was part or like contributed in the mycotic ulcer trial, and is still doing it. So they came from axial ocular infections, which obviously are very virulent and have very high resistance to drugs. And for the second question, I forgot what it was. I think you said.
Yeah, it was. Does the 0.5 value kind of translate well into actual therapeutic synergy?
I don't know for beta blockers besides timolol, because that's the only one that has been shown to work. But I assume it would, because it worked for timolol in the mycotic ulcer trial treatment. So that would translate. But I didn't see any FICI that did work, so I wouldn't be able to use it anyways. But yeah.
Other questions? Are we sure? Okay. Well, let's thank Galini again for this wonderful presentation. Feel free to hang around and ask a question if you didn't want to ask it to the group. We will be hanging out here for maybe about another 10 minutes, and then we will be switching over to the Zoom for Katie Kitzinger's talk. If you want to hang on, I will chat that link to you in case you need it. I will put that in the chat. I just have to pull it up. So I hope to see you there. If not, I will see you whenever I see you at the next talk or in other situations. Thank you all for coming, and thank you for Galini for the wonderful presentation.
Thank you.
Good job.
Yay. I'm getting there to chat this link. That's the link to Katie's talk, if you need it. If I pasted the right one, which I did.
Great job, Galini. That was great.
Thank you so much.
It was nice to hear the whole big story all put together. That was fun.
I'm glad
It
it
really
worked.
did come together nicely. I was very happy with your changes.
Nice.
So thank you for all your hard work. It really came out well.
Thank you so much.
Was that your last big thing of the term, or do you still have some big things to do?
I mean, we still have a paper for the chemistry capstone, but besides that, I have two other classes, which one of them has a final, one of them has a paper. So I still have some work to do, but we're halfway there.
There you go. Halfway done.
Yeah.
Yay. Very good. Well, I'm not going to stick around too long because I know Deanna has to shut this Zoom down in order to start the next one. But it's good to see you, and good luck with the rest of your term, and I'll reach out again before things are done.
Thank you so much.
Okay. Yep, you're welcome. Bye. Have a good afternoon, everybody.
You too.
So I think we're good, and I hope you're feeling good. I hope you felt good about that. It went really well. I think you should be very proud.
Thank you. Thank you so much. Yes. I'll see you in 5 minutes probably.
I will see you in a little bit. Or not. Whatever. See you later.
Bye-bye.